Only a few days of class left. You have a weekend to finish up for whatever classes you've got going, and then you can start focusing on finals. Hopefully you started thinking about them already. Our finals on auspicious Friday the 13th, 7:30 a.m. Remember, 25% of your grade will be by twice as long, cumulative. So don't forget to study everything else we've covered in the class. Don't forget to fill out your ECAPA course evaluation. As of yesterday evening, the response was at about 60%. Okay, so you get up to 85%, everyone gets another 4 extra credit points. So remind all your friends to do that. As far as your midterm exam number 4, today is the last day to come and talk to me or to your TIs about point adjustment. The end of the day or sometime this weekend, I'll try to get all the corrections made. So La Lima might not reflect your course correction yet. I'll get those all done this weekend. Also note that on La Lima, I have changed your exam grades into percentages. So I've scaled them all to 100. So if your exam, it's no longer the points, but the percentage that you got on each exam. That way I can load all 4 exams up into La Lima, and La Lima will reflect your current grade accurately. So it can drop whatever your lowest one is. So that's been updated. And your final review next Tuesday, May 3rd, 5 to 6 in this room. Okay, I think that's all the announcements. Are there any questions over the exam? The review before we get started? All right. Then we're going to finish up with neuronal signaling and get into the last few pathways. So these last 3 lectures, we're going to finish neuronal signaling. So we've gotten through axon, action potential propagation. The only thing we haven't talked about yet is what happens when it hits that synaptic terminal. Once we get to that, we're going to spend the last few lectures talking about some additional membrane-bound proteins and the signaling pathways that they're involved in. All right, so the rest of the time for this course is just signaling pathways. Action potentials. At this point, you probably heard me say it 1,000 times, so you should know this stuff forwards and backwards. You need to know the equilibrium potential for potassium versus for sodium, the resting potential, the threshold potential. You need to know where in that action potential each of the channels we're concerned with, sodium and potassium, are opened or closed or inactivated. And you need to know what each of those stages of the action potential are called. Resting potential, depolarization, repolarization, hyperpolarization. So we finished up on Wednesday talking about why action potentials only travel in one direction down the axon. Just to reiterate, because we covered it pretty quickly right at the end of class when everyone was sort of packing up, maybe not paying attention. This axon propagation and why it only moves in one direction down the axon, that has to do with those refractory periods that we were talking about. And the absolute period where the sodium channel is inactivated, and in the hyperpolarization phase where it is reset, so it's de-inactivated, but it requires a much larger stimulus to get it to fire. So remember, to start an action potential, we need a stimulus that depolarizes membrane. Remember remember that action potentials are first initiated at that axon helix from the passive diffusion of charge that comes from the cell body, that comes from the dendrites. And when that passive diffusion of charge hits that axon helix, there's a concentration, right, of those sodium channels that can then respond to that stimulus and start the action potential. As that action potential fires, sodium is rushing into the membrane. It is flowing in both directions, but only the sodium channels ahead of it down the axon are in a state that they can open up in response to that stimulus. Even though sodium is also flowing in the opposite direction, the sodium channels behind the action potential are in that refractory period where either the sodium channel is inactivated and can't be fired, or it's in the hyperpolarization phase, so it needs a huge stimulus. So because of that refractory period, action potential is only moving a wave down the axon. And then the final piece of this is to come back to those glial cells, those support cells. Talked about back at the very beginning of when we started talking about neurons. Some of these glial cells form a myelin sheath around the axon that serves as insulation. And so when there's these myelin sheaths, what you see are the action potentials jumping from node to node, so the spaces between those sheaths. So you get action potential buildup of those ions, and those flow down, and then you get action potential buildup of ions that flow down to the next node that's free of myelin. And these insulate that stretch of membrane so that can happen. So you get this jumping from node to node, and that's how these action potentials propagate quickly and over long distances. All right, so hopefully you have gone and looked at that video. If not, you should check it out to help explain that axon propagation for review. So that gets us through. Talked about what happens at the dendrites. They're collecting those electrical signals. The cell body is integrating those signals and generating a large enough stimulus that it's going to generate an action potential at that axon helix, send out that signal down the axon. The axon passes that signal to the synaptic terminal, and then at that synaptic terminal, a signal is sent out to the next neuron in the pathway, or to an effector cell. So if we go back and look at that original knee-jerk reflex synaptic pathway that we started this whole section with, we're talking about what happens here. So we talked about what happens to fire this, the propagation, what happens when that signal hits that synaptic terminal. If we look at the TEM of that junction between neurons, so the synaptic terminal on one side, here is a dendrite to the next neuron. Notice there's a tiny little gap between them. In the synaptic terminal side, that terminal is full of synaptic vesicles. So what happens when that action potential hits that terminal that's full of those synaptic vesicles? Notice there are also calcium channels, right? So we're now talking about a third ion channel involved in the passing of the electrical signals from neuron to neuron. So what happens when an action potential reaches that synaptic terminal?
You should know this. What is it?
What happens when those action potentials hit the end of that neuron? What channels are involved? All right, everyone have a chance to answer? Let's take a look. All right. There to give you a hint. It is, in fact, that calcium channel's open. All right, so we're switching from sodium and potassium channels, which are driving that action potential. When that signal hits the synaptic terminal, it then opens up calcium channels. So what happens when those channels open up? So that action potential slows down the axon, hits the terminal, where all those vesicles are just hanging out. Those vesicles are full of neurotransmitters, so that synaptic terminal is just charged and ready to send out a signal. Once that action potential gets there, the depolarization of the membrane generated by that incoming action potential opens up these voltage-gated calcium channels. All right, so these are voltage-gated again. They're responding to changes in the membrane potential. That allows calcium to rush into that terminal, increasing the calcium concentration at the end of these neurons, then affects these secretory vesicles. Once calcium starts to rush into the terminal, those secretory vesicles that are full of neurotransmitters start to get released, secreted into that space between the two neurons, into that cleft. The more you stimulate the end of that neuron, the more neurotransmitters are released across that cleft. Those neurotransmitters move across that very small space between the terminal and the dendrites of the next neuron. Notice that there are receptors on the dendrites specific for those neurotransmitters. So once those neurotransmitters are released, they bind to the appropriate receptor on the dendrite of the next neuron. Those receptors are ion channels. So those channels open when they bind these neurotransmitters. All right, so we've gone from voltage-gated channels to ligand binding channels. They have to bind the appropriate thing in order to open up. So the channels open on the postsynaptic cell, so the one after the cleft. Ions flow into that cell. Depending on what kind of channel it is, it could hyperpolarize or depolarize that cell. If enough depolarization happens from that signal, you're going to build up again that passive spread of ions in the cell body of the next neuron, which then is going to move to the axon hillock and fire an action potential in the next neuron. So when we're looking at the interaction of neurons from the synaptic terminal to the dendrite of the next one, we are going from that electrical signal, that action potential, that's moving down the axon of the first neuron, to a chemical signal, release of those neurotransmitters, that induces opening up of the ion channels, and then converting that back into an electrical signal in the next cell. So buildup of those ions enough, that you get enough of a stimulus, enough of a depolarization to generate an axon, action potential in the next axon. All right, we have to go from electrical to chemical to electrical, so we have to get that signal to jump across cells. Right, those membranes aren't connected. So they can't talk to each other using an electrical signal. There are a number of different neurotransmitters that are used. That's a little bit small, but there are different categories, so biogenic amines, amino acids, neuropeptides. Also notice that some of these are excitatory and some of these are inhibitory as far as their effects on the next neuron. Many of them are used in the central nervous system. Some are used in the peripheral nervous system. Some are used at neuromuscular junctions. We'll take a look at it in a minute. So let's think about this interaction, this electrical to chemical to electrical signal transition. Do this so you can read that. All right, so we're talking about that synapse between neurons. If that channel on the postsynaptic dendrite is a sodium channel, and a ligand binds to that, what's its general effect in terms of generating a new action potential in the next cell? Everyone get an answer?
Yes.
One more second to get your answers in. Take a look. Okay, so ligand binding. The ligand binds to the sodium channel on the dendrite of the next neuron. It's going to open that channel. Opening that channel is going to promote an action potential in the next neuron. So when we open sodium channels, sodium is moving into the cell. It depolarizes, and it's going to integrate in that cell body through that passive spread until enough is accumulated to generate an action potential at the axon helix. All right, so it's helping to move the membrane at the axon hillock from resting potential past the threshold to generate an action potential. So I think I already gave you the answer to this, but I'll let you answer it anyway. So what happens at that postsynaptic membrane when sodium channels open up? I'll give you 1 second to answer that. All right. Take a look. Okay. So it depolarizes that membrane. So some synapses between neurons, depolarize the membrane. And we call sort of the number of synapses that and the sodium channels involved give you the excitatory postsynaptic potential of the next neuron. All right, so that's due to the sodium channels, which are excitatory. So we know what happens here. We know what happens across this synapse. Right, we're going to excite the next neuron with a neurotransmitter like glutamate. We're going to excite that motor axon that goes back to our muscle. What happens in that final part of our circuit? Here we are at a neuromuscular junction. So the neuron is synapsing with muscle cells, effector cells that are going to do something. If we go back and look at our neurotransmitters, keep moving this around, see what's going on. So as I pointed out earlier, some of these neurotransmitters work at neuromuscular junctions, which means that there can be either excitatory or inhibitory to muscle cells. We think about a human. So we have up here vertebrate neuromuscular junction, excitatory. We have the neurotransmitter acetylcholine. So acetylcholine, there's a coenzyme A, and an acetyl group with that coenzyme, and a choline together to form acetylcholine. This neurotransmitter is excitatory to vertebrate skeletal muscles. So if we release this neurotransmitter at our neuromuscular junction, we excite those muscles, your muscles contract, you get that knee-jerk reaction. So the only piece of this that we haven't covered explicitly, although I think I mentioned it early on, how do we get this whole circuit started? Why does tapping on the tendon on your patellum cause this reaction? What kind of ion gates is it stimulating? What kind of general mechanism is opening those ion channels? Everybody remember their stretch-gated ion channels? The stretch-gated are a physical opening, stretching of the tissue and the membrane that physically pulls those channels open. So when the doctor taps your knee, it's stretching the tendon, which is opening up these ion channels, which is allowing sodium into the cell, which then passes the threshold, inducing an action potential in our dorsal root ganglion neuron here. All right, to generate those action potentials in our first neuron. All right, we're already partway there. We have our stretch-sensitive gated ions on this end. Generates an action potential in our first neuron. Stimulates the release of glutamate, which is an excitatory neurotransmitter. That is going to depolarize the next cell and generate an action potential in the motor axon. That action potential is then going to stimulate the release of acetylcholine at the neuromuscular junction. That acetylcholine is going to cause the muscle to contract. All right, so that's So look at a simple system. I talked at the very beginning about variations in neuron structure and function. What about a neuron that looks like that? So we have this whole tree of dendrites, the cell body here, and an axon. When will this cell fire an action potential? All right, so we talked about a pretty simple circuit. We've mostly talked about sodium channels opening and closing. What about potassium? So in our synapses, if the neurotransmitter, the ligand, interacts with the potassium channel, what's going to happen to the cell membrane?
That's not how you don't get braces. You get it
All right, everyone get their answers in? Let's take a look. All right, I think you guys got this figured out. Are you trying to ask about polarization and depolarization yet? All right, so when potassium channels open up, it's going to hyperpolarize that membrane. It's going to make it harder to depolarize the membrane enough to send a signal. So it's going to inhibit that particular neuron from firing. All right, so the concentration of the potassium channels and the synapses, because potassium channels are inhibitory, they make it harder to fire off an action potential when they're opened up at the dendrites. That gives you an inhibitory postsynaptic potential. So what happens when you put these two together? We have excitatory potential and inhibitory potential. And if you look at synapses between dendrites of one neuron and the terminal of another, the red is inhibitory synapses, green are excitatory synapses. And depending on where the signal comes from, you have some combination of both going on. So it's not always just a simple, you get a signal from one neuron that says go, you depolarize, and you send the signal on. These neurons can be complex networks of signals that are both, some are exciting, some are inhibitory. So you have to get enough of those excitatory to overcome that inhibitory potential to fire off that neuron. So you can just keep that in mind. So if you think about when this cell will fire an action potential, we don't know, because we don't know how many of those synapses are excitatory, how many are inhibitory, where those signals are coming from. Right? Biological system, so it's complex. All right, I think that is it for neurons. Are there any questions about action potentials, synapses, before we move on to the other signaling pathways? With the last few minutes of class, I guess the last 15 minutes of class, we're going to start talking just introductory about the next set of pathways we're going to talk about. Question?
What's the answer to this question?
Can't tell you, because we don't know how many of those synapses, those dendrite synapses, onto something excitatory, and how many are synapses that are inhibitory.
When it's more excitatory and inhibitory inside?
Well, so you have to have enough signals coming in, because the signals are synapsing from different places.
Yeah.
If you're only getting signals in the excitatory, then it's going to fire. If you're getting some combination of signals, then it depends on which one is winning, right?
So if excitatory is winning,
You'll be able to get past that threshold value. All right, it's all about getting past that threshold value.
is it also like time dependent? Like if it's too far apart, kind of.
It is also time dependent, right? So when these dendrites get a signal, they open up the channels, that's transitory. So you have to have enough signal build up in the cell body. But remember, there are also background sodium-potassium channels going on that are going to equalize things. And the sodium channels are leaky. They're going to equalize things back to resting membrane potential over time. You have to have enough of that come in the right time frame to get that threshold. So it is time dependent. Any other questions before we move on?
Yeah, one more thing. Back in the previous slide, what was DRG?
DRG is dorsal root ganglion. So that just describes that ganglion that is coming from your peripheral system to the spinal cord.
Do we need to know that and draw that in?
Sorry?
Do we need to know that and draw that in on a diagram?
No. You do not need to draw that in on a diagram. Any other questions? Okay, see a hand up the back, Tony.
So when potassium is low enough, so you're hyperpolarizing, right? And in that state, it's also inactive. If we're still under the category of IPSP, does it stay active until it reaches resting potential? Then the ball drops, and now it's just closed?
So when we're talking about inhibitory and excitatory, we're just talking about sodium and potassium channels at the synaptic cleft, so the dendrites.
Okay.
So these aren't firing action potential. It's the sodium channels along the axon that are. So we're simply talking about them being open or closed. And the potential, so your excitatory or inhibitory potential has to do with how many of those channels there are in the dendrites. Right? So how quickly can you generate that charge if they receive a signal? Yes. Yes. Did I have my box drawn wrong? Right, so let me draw it on the board to clarify. So the question is about the absolute and relative refractory period. So it's inactivated here, and it's not deactivated to here. Yeah?
Yeah.
So this is the absolute refractory period, and that would be the relative refractory period. I had my boxes slightly off on that diagram. I'll fix it. Thank you for pointing that out.
So can a channel inactivate and deactivate even if it doesn't? So like if there's only potassium being opened up, doesn't still do the whole inactivation.
So it's the sodium channels that do the inactivation, deactivation.
Oh.
Potassium channels just open or close. Okay? Clarify that? What about the yeah, these are the two refractory periods, absolute and relative. And these keep action potential from going backwards along the axon. You can generate a second action potential here, but you have to have a huge depolarization stimulus. So it makes it much more difficult. Generally, when you're talking about the ions that are diffusing out from an active action potential, membrane depolarization is not enough to get past that extra amount needed to get to the threshold. Sodium channels inactivate at the peak depolarization, and they de-inactivate at the peak hyperpolarization. Any other questions I can clarify for you on action potential while we're here? No other questions? All right. I hope you all appreciate action potentials as much as I do. I think they're fabulous. We're going to move on, though, and talk about some additional mechanisms of signal transduction in the cell. These signal transduction mechanisms are all driven by membrane-bound receptors. What we've been looking at with action potentials and neuron firing, these are all ion channels. So in our circuits, we're talking about sodium and potassium channels, calcium channels, along this entire pathway that are driving that electrical signal, and that electrical to chemical to electrical signal. The last two types of receptors and signaling pathways we're going to talk about are G-protein linked pathways, G-protein linked receptors, and enzyme linked receptors. When we're talking about signal transduction, we're talking specifically about the cell detecting something outside the cell and turning that into a cellular signal. So these signal transduction pathways, generally they all have to start with a signal outside of the cell. So the cell has to some way detect that signal. It signals through a membrane-bound receptor. So the receptor interacts with that signal on the outside and changes something on the inside. And then that starts a signaling cascade that results in some sort of cellular response. So signal transduction is going from outside the cell to inside the cell, inducing, so a signal outside the cell inducing a change inside the cell. So rather than ion channels, which are allowing the passage of ions and change and producing an electrical signal, changing that membrane potential, we're talking about receptors, again embedded in the membrane, but they're going to bind some molecule outside the cell that's going to change their conformation inside the cell and allow something to happen. So these are not channels. Nothing is moving across the membrane. What we have are proteins embedded in that membrane. Remember, we talked about how this happens. We have bits of protein on the outside of the cell, bits of protein on the inside of the cell, so that when the part of the protein outside the cell interacts with the proper ligand, molecule, or signal, it changes shape, allowing the inside part of the protein to interact with proteins on inside the cell and start that signaling cascade. So these types of signaling pathways can result in multiple cellular responses. They can function to alter gene expression, which means that signaling cascade has to get from that receptor protein through the cytoplasm into the nucleus and alter what's happening with transcription. And this is a slow process. Just think about it if you are altering transcription, right? So You have to get into the nucleus first, then you have to alter that machinery, and then the whole pathway down, you have to have time for the whole pathway downstream to be altered. Translation, protein modification, etc. A faster pathway is when these receptors have a cascade that alter protein function. So there are already proteins in the cytosol. When that receptor binds a ligand, that just alters the proteins that are there and either turns them on or off. So that's a fast response as far as the cellular response. We're going to talk about, for the last few lectures, 3 specific protein-mediated signal transduction pathways that lead to this altered protein function. So we're going to talk about the adenyl cyclase pathway, the phospholipase C pathway, and the RAS-MAP kinase pathway. And those are the last 2 pathways you have to learn for this class. All of these pathways are similar in that they activate a protein kinase. Adenyl cyclase activates kinase A, phospholipase C activates a protein kinase C. The RAS-MAP pathway activates a MAP kinase. All right, so what's my next question to you? What's a kinase? What does a kinase do? I heard it. It phosphorylates proteins. So kinase is simply a protein enzyme that phosphorylates a target protein. And that phosphorylation of the target protein is going to change that protein's activity. So we have a protein that is currently not active. Through interaction with that activated receptor, it gets phosphorylated from ATP. So it pulls the phosphate group off of ATP. And that activates that protein. So that's a fast response. And that's all due to protein kinase activity. Excuse me, in each one of those pathways. And then to reset that signaling transduction pathway, that protein that's active needs to be dephosphorylated, and that's due to a class of enzymes called phosphatase. The kinases phosphorylate proteins. Phosphatases pull that phosphate back off. So that we can get this rapid signaling by phosphorylation response. We'll stop there. We'll pick up with those last 3 pathways on Monday. Feel free to look them over the weekend. Get ready. Looking ahead.